Okay. Good morning, good afternoon, and good evening to all those who have joined us. We welcome you. And could we just queue up our slides so we can begin? And thank you very much for joining us. This is a very important meeting for us of those who are passionate about biorepository. I think last year, one of our speakers, Anthony Risk, said it very well. If this were easy, it would have been done a long time ago. And this has not, excuse me, this has been a challenge because, sorry, these are challenges I think we should be able to parse out and move forward together. And it's just something that not a single individual or group can do. So I'm really pleased that you're here, and we will just get the slides queued up and we will get started. So what we like to do is to give you an overview of what we're doing. So our talk today, our meeting today is about, partnering for the response to the pandemics of the future and biorepositories for the public good. As I said, this is something that just has not been done as readily, easily for various reasons. And our goal here is to gather together people, what I call the coalition of the willing, and to be able to then tackle each and every one of the questions that we have in order to be able to do this. We're very grateful for our partners. We are underlying this is we have the Bill Melinda Gates Foundation, who have supported a number of our projects, as well as the EU Commission. And so these are our partners: the Global Health Network, PATH, the Colorado School of Public Health, and ReCoDID. We thank them for their support. Here's some housekeeping. This webinar is being recorded and it'll be shared on the Global Health Network platform. So please use the chat box to introduce yourself and to post your comments. And please then use the Q&A function to post your questions so that we be sure that we can triage your questions and answer them as best as we can. On the Zoom controls, because we have expecting high number of participants, your camera, microphone has been turned off. So use the chat function for any technical issues. And then when you have such technical issues, please select the Global Health Network team to help you resolve it. So our concept has been to empower individuals and regional sites to collect and retain samples for advancing research, diagnostics, vaccines, and therapeutics. What we wanted to do was also coalesce as a federated virtual biorepository partnership where members together find solutions to access high quality materials for public health needs and to collect what WHO, you will soon hear on what they call the biological materials of pandemic potential, because these are very important for early on when some agent reemerges or emerges, to have the adequate samples and types pre-characterized in order to quickly facilitate diagnostics and research. And then our goal is to really have a very trusted and sustainable system that will provide equitable access, ownership, recognition, and benefits sharing. The approach that we're doing is 3 steps. Last year, we had an introductory workshop, and this year we are developing working groups. And these are the 3 working groups towards the end that you will have discussions on, and you would then be asked to join us to sign up for any of these working groups. And then we want to operationalize, which is the future implementation in volunteer sites, harmonize sample sharing mechanisms, provide access to benefit sharing. And also we want to create a probably unique in these days, a sustainable and equitable business model. So today we're going to start with the speakers, and I'm going to go through this list. You can take a look at the list, and I will introduce them one by one as they come up to speak. First and foremost is Dr. Tony Moody from Duke University, and he will speak on the conceptual models for biorepositories. So Tony, you can take it away.
So I'm going to run through this very quickly. Like May said, I'm going to talk about conceptual frameworks for thinking about biorepositories. This is something we've been doing a lot of inside the CREED network, thinking about how we can operationalize this for our network and for other networks. So I'm going to start out with a few assumptions. Number one is samples have value when they're used. They're not like wine. They don't just accumulate value over time. And so we really do want our samples to be used, but we also work very hard to get those samples. We want to make sure that they're not wasted. And we want to make sure that our hard work is recognized. This is really important. We're all, needing to get recognition, promotion, tenure, all of those sorts of things. And so, we want to make sure that this is going to work for us as well as for other people. And of course, we all have this vision that this is what science is like and everybody's sitting around smiling. And of course, it's all very collaborative. But from the outside, it can often look like this, that we're just sitting on a treasure hoard and people are going, why won't you share your samples? And of course, there are lots of reasons for that. And we'll go through some ways of thinking about how we can potentially operationalize this. So I'm going to present 5 models for specimen collection and sharing. 2 of them are distributed and 3 of them are centralized. And combinations or hybrids of these are possible. And really, this is just a starting point for discussion. This is not meant to be some set in stone kind of idea. I really would appreciate feedback if people have thoughts of how this could be modified. These are conceptual frameworks for talking about biorepository models, and all assume that people are collecting samples of interest both to them and or their collaborators so that people would actually want to access them. So the first one I call the private collector model. This is one where a collector may wish to share information about their collection on a limited basis, but mostly they're interested in collecting these things for their own purposes. And they may be willing to have someone act as an information broker, but access would have to be negotiated. This is actually the model that most of us are in most of the time with our biorepositories. We're collecting things, we're using them. Occasionally, we will share information. This is obviously the most restrictive model and really something that I think this working group is trying to get past. The second one I call a museum model. That is one where you've got a collection, but you're willing to share a public catalog about the collection. But just like a museum, it doesn't mean that you can just walk in and take something off the wall. You can see it. And if you want to use it, you would have to go through a negotiation process. So a coordinating center could act as a collector of catalogs and basically provide a view of catalogs across sites, but it wouldn't actually guarantee access. Access would have to be negotiated. And this is this is one kind of virtual biorepository model. And in fact, we might think about a hybrid model here where you would actually have based on your status, you would provide a limited view. You might be able to see the types of samples, but not the location. You might be able to see that someone has a given category of specimens or about a specific disease, but then in order to get a different view, you would have to go in and negotiate with them to get a better view of what is in their biorepository. There's a model that I call the bank model. So, and this is sort of taken from the Swiss banking idea of groups would deposit a portion of their collection to a central facility, and that facility would hold the physical items, but the group that deposited them would retain control, very much like a safety deposit box. And access, again, would have to be negotiated. Now, this is a central biorepository model, but it's important to notice that the locus of control retains or stays with the people who deposited the materials. A model that we've been having a lot of conversations about is something I call a bookstore model, where this is an idea where groups would deposit a portion of their collection to a central facility, and the connections would remain in place with the group that is providing the materials. And the originator can set a price, and you can define price very broadly, but just think of it as the conditions for release. And you would actually get more rapid access if you meet the price. If you want to negotiate a different price, you would have to enter into a negotiation with the originator. And this is a kind of central biorepository model where you may be able to have faster access to something, but you would retain that connection to the people who originated the specimen, which may retain value. And then the last one I call the thrift shop model. That is, groups would deposit a portion of their collection to a central facility, and then that biorepository would set the conditions for release. And this would be the one that would provide the greatest and most rapid access, but just like a thrift shop, connections to the originator may be lost. And I think a lot of times when people think about biorepositories and sharing, they think of two extremes. They think of either the private collector model or this thrift shop model, but they don't think about the things that are in between. And so I think it's really important that we have a conversation about possible solutions that come in between. Now, there are a lot of questions that these models raise. Because of course, are specimens still valuable if individual, for example, identifying information is not available? Sometimes yes, sometimes no. And so I think the model that you choose will have to be reflective of the value that the specimens have when that information is connected or lost. And really, how much information is needed to retain specimen value? And again, that may vary depending on the experiment. And then, of course, on the right side, who benefits? And there are lots of things we need to think about, right? The Nagoya Protocol is there in order to prevent just, willy-nilly sharing of genetic information, often used as a shield to say, well, I can't share because of Nagoya. We could have a conversation about that later. But there are other things. There's the intellectual property issue. There's the issue that some of us need to worry about promotion and tenure, or maybe you need to have recognition from a governmental entity. And then the last one, who watches the watchers? And, how do we set up a governance structure to make sure that the biorepositories are doing what we need them to do? Now, it's important to understand that possession in a biorepository does not equal permission. Just because you hold the specimen doesn't mean you have the authority to do anything that you want with it. And of course, one way we can think about this is the idea of a legal trust. That is, you may have a grantor, the person giving the thing up, and the protector, the entity that is overseeing whether or not that can be released, and then the beneficiary who might receive that kind of information. And we're very comfortable with this idea from human subjects work in that there are IRB or ethics protections, that the participant is providing specimens, but that the IRB is providing oversight before the researcher can receive those specimens. And of course, if you're in a biorepository situation, there may be multiple layers of this. You may have to have that degree of oversight before something goes into a biorepository, but then you may need to have a process within the biorepository to make sure that everything is being appropriately dealt with before specimens are released. And of course, when you're talking about animal specimens, a different set of regulations may apply, right? You may have other regulatory authorities or regulations like CITES that apply. And so you have to think about what is going to be necessary for oversight before you deposit something. And then there are some final considerations, such as biosecurity, like who has access, who should be able to see what's there. Do we really want to provide global access to say, oh, there's a collection of smallpox specimens at X location? Is that really wise? So do we have limited viewing rights? How do we support biorepositories? Everybody wants to have a biorepository when an outbreak occurs. Nobody wants to pay for them in interpandemic periods. And so thinking about how we support the infrastructure is key. And then, of course, there's the political angle. How do we navigate international politics? Because, of course, not every country wants to share with every other country. And we do have to think about these things, even if they are things that we would prefer to not have to think about. So with that, I'll stop. Hopefully I came in on time, and I will turn it back over to May, who can introduce the next speaker.
Thank you very much. So the next speaker, thank you. That was very insightful and a lot of things to think about. The next speaker that we have is actually a recorded recording from WHO. Dr. Iaona Ghiga will explain the WHO BioHub that recently began. And this is something that I think we are fortunate to have and to think about synergizing with. Could you please go ahead and share their video?
Good morning, good afternoon, and good evening, everyone. I'm very pleased to be with you today via this video. And I would like to thank the organizers of this meeting, and in particular, May Chu, for inviting us. I will talk to you today about a new initiative that WHO has been developing in the past months. And this initiative answers a need expressed by our member states. It's called the WHO BioHub system. And in my short presentation today, I will give you an overview of what we are hoping to achieve, how we are going about achieving it, and where we are presently with developments. So as you know, when a new disease emerges, it usually starts with an unusual cluster of cases. And as part of the first efforts, it's very important that we identify the causative agent, that this pathogen is analyzed, that the results are shared as broadly as possible so that risk assessments can take place, and also that work can start towards development of medical countermeasures such as diagnostics, vaccines, and therapeutics. The pathogens may arise in different settings and country capacities vary. So it may be the case that sometimes the country where the first identification of this novel pathogen took place does not have the necessary means to do an in-depth analysis or to be able to quickly share at scale the pathogen with more labs around the world. And this sharing at speed and scale is important because it would increase the efforts to perform rapid analysis by more labs at the same time, which would enable the outbreak response and will lead to more timely actions to curtail the outbreak. So the WHO BioHub system is looking to help with all this, and it aims to promote this timely sharing, to facilitate rapid access to biological materials, and finally to ensure fair and equitable access to such products by all countries based on public health needs. The system will only apply to biological materials with epidemic or pandemic potential, and it is not meant to replace any existing systems or sharing pathways. It is providing an additional service for member states and other stakeholders that wish to use it, and make sure that when a completely new pathogen appears, there is a clear pathway that can be activated to facilitate its sharing. As mentioned, we are working in accordance with the WHA resolution, and on this slide you will have more details about the text of this resolution, so I won't go into too much detail now. When it comes to the approach to conceptualize the system, what's important to note is that when a new pathogen appears, member states can voluntarily share the biological material through the use of standard material transfer agreements with the BioHub facility. For now, we have only one such facility. It's a BSL-4 lab. It's located in Spitz in Switzerland. And once the material is in the facility, the BMAP is analyzed and all preparations are done so that it can be rapidly shared with the other requesting entities, which we refer to as qualified entities. Again, this further sharing will be done using standardized documentation, so via SMTAs, and the aim is to enable more knowledge about this pathogen and facilitate development of needed products. The system is anchored in 10 guiding principles, and they pertain to the voluntary nature of this initiative. No one is obliged to use it if they don't want to. It will be a system that will be guided by equity and fairness. It should enable a timely response, be characterized by transparency in its terms and conditions. It should ensure collaboration and cooperation, as well as acknowledgement and co-authorship of all the scientists that are involved. The work undertaken should be done under best practices for safety and security and be consistent with applicable law, as well as ethical regulations, norms, and standards. And the system, of course, should be established and managed with longer-term sustainability and maximal preservation in mind. In developing the system, we are taking an iterative design approach. And our work is structured on two streams. One is what we refer to as a pilot phase, and the other one is a more extensive system design endeavor. The pilot phase is used to really look at the things that we can do operationally so that we can make progresses fast and fit for purposes. And we are using SARS-CoV-2 as a test BMAP to enable sharing. This sharing is done only for non-commercial use in this pilot phase. On the other work stream, we are engaging in a series of technical consultations, and what we are trying to achieve is gather views from all stakeholders on critical issues that have to do with designing the wider system. So this will also aim to gather considerations on the terms and conditions that would have to do with commercial use sharing. And on this slide, you have an overview of all these consultations, all the themes that the consultations are addressing. Two of these have already taken place. The one on handling research results and appropriate recognition of contributions. This was followed by a consultation that focused on genetic sequence data handling. And this week, we are going to have another one focusing on intellectual property considerations. So we hope that you will attend it. For all these sessions, we place information on the WHO BioHub webpage. So you will be able to find there the background documentation, the slides that were used in the respective meeting, as well as the recording of the meeting itself. Next year, we will continue the technical consultation with one that is dedicated to access and benefits considerations. And we will, of course, be reporting to our member states on the inputs that we will receive. So in closing, I would just like to reiterate that the BioHub is a voluntary system. It is meant to be an additional service to complement existing sharing pathways. And we really look to learn from all the initiatives and biorepositories in this space and leverage all existing available efforts. We are co-developing this system, and we are interested in hearing from all stakeholders on suggestions because we really want to make this a truly valuable service so that we are better prepared for responding to health emergencies that result from infectious diseases. Thank you, and please visit our website and don't hesitate to get in touch with us. Best wishes on behalf of all the team here at WHO. Goodbye.
Thank you very much. So that was introduction to the WHO BioHub. And of course, since it's recorded, if you have questions, please post it in Q&A and we will try and address them and send them forward to Dr. Ghiga if we can answer those. Next, so that just illustrates some of the bigger things that are happening. The next is much more practical talks that we'll be giving you, and that will be led off by Dr. Roger Peck of PATH, and he's going to give an overview of the setting up of a biorepository as COVID-19 epidemic unfolded. Roger, do you want to take it away?
Great, thank you so much, May. Let me get my screen shared here. Good morning, good afternoon, good evening, everyone. Today I'd like to share briefly with you our experiences establishing a COVID-19 biorepository. For those of you who are not familiar with PATH, PATH is a global nonprofit working to accelerate health equity so that people and communities can thrive. Located in Seattle, Washington, we were uniquely positioned to respond to COVID-19 as the first identified COVID-19 case in the US was in our region. PATH has decades of experience adapting and developing diagnostic tests for manufacture and deployment in resource-limited settings. We know that diagnostics are central to controlling diseases, and that outbreaks and epidemics place additional stress on fragile health systems. Information is critical to informing individual and public health decisions, and the traditional development timeline of 3 to 5 years for medical devices wasn't acceptable for the COVID-19 epidemic. To bend the curve, we knew it would be a cooperative effort with multiple players bringing knowledge and assets to the table. If our experience in global health has taught us anything, it's that partnerships are critical. We relied on our existing networks to form the Washington COVID-19 Biorepository. The repository is founded with the framework of a governance plan with principles for safe handling and ethical use of specimens. The repository is overseen by a governance committee. We have leveraged previous examples of our templates for standard operating procedures and legal agreements. Our collaboration with contributing institutions includes donors, medical providers, state public health labs, academic institutions, and private labs. Each repository relationship is grounded in ethics, continual learning and improvement, and most importantly, the sense of urgency to leverage knowledge and resources to advance safe and effective technologies that address the needs created by the pandemic. Additionally, the repository is driven by equitable distribution of repository materials and commitments to underserved populations by the recipients. We reached out to our global network of diagnostic developers to identify their needs and determine how the Washington COVID-19 Biorepository could advance their work. The repository is positioned to support developers of rapid point-of-care tests best suited for resource-limited settings. The repository houses specimens appropriate for molecular, antigen, and antibody test development. While challenging in the earlier days of the epidemic, we aim to collect many high-quality specimens and link the specimens to useful data. Consistent with PATH's mission and stewardship of the specimens, the repository materials are available to qualified researchers for free, though recipients pay for shipping costs. Application is made through an online request and processed in a timely manner. Recipients are required to sign a material transfer agreement and that the institutional commitments are codified. Such commitments include orientation to tests, orienting tests to global markets and manufacturing products at scale to address the demand. We continue to add materials to the repository and promote the availability of materials in the repository. To date, the repository has been going for less than 2 years. We've received 63 submissions from 12 countries. 45 of the submissions were approved, and we've identified that 6 emergency use regulatory pathways were supported by specimens from the Washington COVID Biorepository. We hope that the repository continues to support the needs of device developers and that the infrastructure can be a model for other repositories as needs arise. Thank you so much, and I'll turn it back over to May.
Thank you very much. So our next speaker had to step out for a little bit, so we will share her slides with you.
Hi everyone, and thank you for this opportunity to tell you more about the African Data and Biospecimen Exchange. I'm Nicki Tiffin. I work at SANBI at the University of the Western Cape in Cape Town. And this program has been funded by a recent award of a Calestis Juma Fellowship from the Bill and Melinda Gates Foundation. The African Data and Biospecimen Exchange will be an online platform that facilitates the equitable and transparent data and biospecimen sharing of resources generated in Africa. There's some very significant obstacles to data and biospecimen sharing of these resources. For example, there's a reluctance to send resources out of Africa to centralized repositories. And there's also a fear of being scooped or excluded from the analysis of new data and biospecimen sets that are generated. Sometimes the data can't actually be ethically shared if there's not the correct informed consent in place from participants, and also the national legislation, health acts or privacy acts, may make it very difficult to ethically share or legally share data outside the continent. And then finally, African researchers are often struggling to keep their research sustainable. And so the value of biospecimens and data sets are really enormous to the researchers to keep the research enterprise up and running. The platform will directly connect data generators, the people who are generating the samples and the data sets, to data consumers, the researchers who want to use those resources. There won't be any centralization of the resources, so we're not setting up any kind of repositories of biospecimens or of data, but rather the platform will expose the metadata about the data sets and biospecimen collections. And then the data consumers will be able to search through the metadata and identify data sets or biospecimen collections that they would like to use. And then they can connect directly with the people who've generated those resources, and there will be the infrastructure on the platform to set up various kinds of agreements, maybe agreements for sharing, data sharing or biospecimen sharing agreements, also agreements if parallel collaborations are going to take place, collaborative agreements, and then importantly also benefit sharing agreements. So to help with research sustainability, for example, researchers in the global north might be able to contribute something towards the efforts of the African researchers who have generated the data and the resources. They might offer to host a student or a postdoc, or to help support a master's student, for example. So all of these capacities to set up different types of agreements will be in place on the platform, and they'll be able to be tailored to the direct partnership between the data generator and the data consumer. So they'll also be flexible for the different kinds of legislations and restrictions for the different countries in Africa. So this is just a general overview of what we have planned and what will be coming together in the coming years. And I really welcome your comments and discussions. And thank you for the opportunity to share with you our plans for this exciting platform. Thank you.
So Dr. Thomas Jaenisch would talk about the importance of harmonized data and specimen sharing. So Thomas, your time.
Thank you, May, and good evening, good morning, everybody. So I'm going to share the approach ReCoDID has taken. So ReCoDID stands for Reconciliation of Cohort Data for Infectious Diseases. And it's a consortium funded by the European Commission that I coordinate out of Heidelberg University. And I also, now I'm based in University of Colorado, which is a partner in ReCoDID as well. So why cohorts? Cohorts really offer a unique opportunity for sequential data and samples to be linked. Cohorts represent a significant investment of funding, obviously, for the underlying research infrastructure. In ReCoDID, we try to marry the clinical epidemiological data with the high-dimensional lab data. We support multi-purpose cohorts, and we support the quality of metadata and quality of sample curation. So in a nutshell, so these are the work packages, and as you can see, governance and the searchable portal for cataloging is in the heart of ReCoDID. And in this work package 5, which is led by Aix-Marseille University, by Xavier de Lamballerie, we are focusing on the development of a local governance model and a searchable portal for decentralized cataloging of data and biobanks. So we are not sharing any samples there. It's about data and the high dimensional results from samples. And here really the focus is on the local component. So we have other projects that are kind of precursors to this one, like EVA-G, the European Virus Archive, that were funded by the European Commission as Collections Without Wall. We are strongly convinced that the governance needs to be thought from the bottom up, not from the top down, and there needs to be a benefit for local cohort PIs in Latin America, in Asia, and Africa, so that they can continue to produce high quality biological samples that are well characterized clinically and that are, of course, stored and managed under the best conditions. And in order to support this local bottom-up structure, we have invested in creating algorithms or flowcharts that represent the local decision-making structure for selection and sharing of samples. So this is an example for a site in Colombia, University Industrial de Santander. And it's within the Zika Alliance Consortium, so it's about sharing Zika samples, but other sites have now also been mapped the same way for in our partner sites in Venezuela and Peru. We have done the same exercise, and it's really to try to map and represent this local decision-making structure so that people in these local sites feel that their their decision as well as their players are being adequately represented in the biorepository. Then the other side of the story, of course, is the searchable portal for cataloging. And that is the European COVID-19 data platform for ReCoDID. It's hosted by the European Molecular Biology Lab, EMBL. And so there we have the SARS-CoV-2 data hubs and COVID-19 portal that are complementary, that allow data upload, that allow also for local users to then have added analytical tools once their data is uploaded there that they can use in order to analyze their samples. So here is where clinical epidemiological data and sequence data then will be deposited. So that's our angle. So we are taking an angle that emphasizes the cohorts as really valuable resources, that emphasizes the local governance structure, and that emphasizes the linking of the clinical epi and the high dimensional lab data. And with that, I thank you for your attention and hand it back to May.
Thank you very much for that, Thomas, and you're right on time. Thank you. So the next speaker will be Dr. Judith Giri, and she will give a summary from last year when we had launched a public poll and also did some questionnaires of small and large industry manufacturers for what they were doing in the sphere of biorepository. So I'm going to let Judith take it over.
I will walk you very quickly through the results of a survey and interviews we conducted to identify what are the gaps and barriers to access to specimens. This is what we did first was a survey that was open to the public. And the purpose of it was to understand what respondents would consider as the benefits of a virtual biorepository, and also to identify what are the greatest barriers to access to specimens. We also conducted interviews, as May just mentioned, with representatives of the different types of organizations, from diagnostic companies to nonprofit organizations and government academic collaborations. And this is a quick overview of our results. We had very good representation, broad representation of participants. Geographically, about 30% were from Africa, about 20% from Latin America, from Europe, Asia, and so on. We also had very good representation of the roles and types of organizations that participated. This was from the survey results. I will go into a little bit more detail what those look like, but just wanted to point out two things. One, that we asked the panelists of if they would be interested in contributing to some well-characterized samples for reference materials and panels. And about 38% said that they would be interested. And it was also very interesting to us that when we asked people about the Nagoya Protocol, about benefit sharing, majority of the respondents, more than 50%, did not know if their country had policies in accordance with the Nagoya Protocol and if or how it applied to them. So that's something important for future follow-up. We asked about the benefits of participation, what type of functions and resources people would find interesting, and what opportunities for collaboration would be there. And this is graphically showing you what people thought were most important to them. The features of greatest benefit that respondents identified for the biorepository was standardization, standards, for procedures, SOPs, standards for templates, for consents, standardization of both processes and procedures involved in the biorepository. The other very important aspect they identified was the need for coordination, help with coordination of logistics, from accesses, managing the access as well as the distribution of specimens. Inclusion in a directory or catalog was considered important. And also people thought that it would be very helpful for them to have access to IT tools and expertise and biorepository expertise in setting up their biorepository operations. When we asked investigators what would be most important, most beneficial to them or to their institution, the highest, more than 80% of the participants thought that networking, opportunities for networking, both locally, internationally, and opportunities for collaborations would be most important to them. Next was capacity building, building capacity for biorepositories for infectious diseases, opportunities for training, as well as opportunities for funding and recognitions and trusted source were next. So the standardization standards for SOPs for the biorepository, coordination of logistics, as well as the networking and capacity building came up as most important. We have also conducted interviews with a lot of different organizations from large diagnostics companies to small companies, nonprofit organizations, and of course, they all have different needs and priorities, but what came across that for everyone, it was important. The quality of specimens and the completeness of data was one of the major concerns. Access to needed specimens and specimen types, and availability of well-qualified specimens for panels and reference materials. To give you a couple of examples, for example, for the small diagnostic companies, identifying reliable sources, especially early in outbreaks, is their greatest barrier. The quality of the specimens and occasionally the quantity of specimens available and having sufficient data accompanying the specimens was considered another very large barrier for them. Access to characterized specimens and clinical data in a timely manner in an outbreak, availability of specific types of specimens needed depending on pathogen stage of outbreak was common to everybody we interviewed. Negotiations, that legal hurdles were brought up as another delaying factor. For those who are working with in low and middle income countries, issues of logistics, of biosafety and biosecurity, and the differences in regulations in different countries came across as a hurdle. In some cases, data access restrictions as well. And then the nonprofit organizations were also brought up what the advantages would be of coordinating efforts and having regional support for the biobanking. So the overall cross-cutting issues that came out from all the survey and from the interviews, where these were the most important: identifying and accessing specimens, having trusted sources, access to needed specimen types, having specimens accompanied with good data. The quality of both the specimens and the data was very important. Need for standardization and for collecting complete clinical data, and then building a sustainable infrastructure that will provide benefits to participants and partners, the benefits and sustainability. I put up here our address for if you want to contact us for any questions or to follow up with us on the biorepository. Thank you.
So thank you very much. I think, you know, this gives you a view of all the complexities and things, and many, many hours went in. Thank you to the speakers for providing their insights. And I'm sure it has, you know, raised a lot of questions. So we're gonna just take a deep breath and we're going to go into the panels, and we're gonna start. We'll be having 3 different panels. These panels are, the first one will be panel 1, be guided by Dr. Jorge Munoz from the US CDC, specifically on what types of specimens and data should be collected from those hard to get, rarer samples like dengue, Zika. Some of those have access to dengue more than Zika, of course. And then to those that are abundant with access to, as in COVID-19, what we're going to do with all those samples that's not been collected, that be guided by your comments. Panel 2 will be guided by Dr. Thomas Jaenisch, and he will especially identify solutions to barriers that we have now in accessing specimens and data. And panel 3 will be kindly guided by Dr. Christine Pratt, who specifically on the types of benefits there are for users and contributors, and how do you sustain it? And Dr. Pratt can speak to that wisely because she is the lead, one of the leads of the EVAG, which has been very successful in exchanging biological materials. So we're going to go ahead and start with Dr. Munoz, and he will begin the discussion with you. And I'm going to go ahead and put him on the screen. All of you, unfortunately, this is not a highly interactive system. If you have questions or you have things and ideas that you want to say, please post your questions in the Q&A and we'll try and address them as quickly as we can. Meanwhile, he will also read some of the questions to you that we've posed. Jorge, would you like to go ahead and take that?
Okay. Yeah, so what I have done here is to get the discussion started is to briefly refer to some of the lessons learned from dengue and Zika. Some of the common issues between those two viruses, and the reason I have them together is because there is a RT-PCR and IgM detection during disease window. Okay, so that's an important topic to cover in terms of samples for collections. And their samples are optimal in this area. And the longevity and specificity of the antibodies, particularly IgG long-term detection in various flavivirus backgrounds is of great importance. There's some specific issues in terms of persistence of Zika virus, RNA, pregnancy status, sample type. Additional sample types are important for Zika. Zika was found in semen and other body fluids, and of course, dengue virus serotype representation is important. So this boils down to acute and early convalescent samples from virologically confirmed cases and prospectively collected samples with defined primary, secondary Zika status. For COVID, it's a new field for me. And I'm glad that Roger presented before me. But this virological detection, serological diagnostic testing issues emerging and rapidly changing. The RT-PCR and antigen detection testing. So those are important samples for anything that has to do with test validations, and detection of variants has become important. The example of the S gene target failure issue, important to compile those kinds of samples throughout the pandemic, and the longevity of detection in nasopharyngeal swabs is of great importance. And the serological diagnosis testing has been also evolving with time. Longevity and specificity of the antibody testing has become important in collections to representing our collections, and the prospectively collected samples from naturally occurring infections and vaccines through time. Those are the kinds of samples that people are after, and so this boils down to nasopharyngeal swabs from disease window throughout various periods of the pandemic, and the prospectively collected samples with defined serological results. So what I'm going to do next is to not sure why this is not. Yeah, is to open the floor for dialogue and hear from your opinions in the topic.
So maybe I can just get that kick started. I think that the minimum quality samples should be some of the things that we have to have a minimal data set, and that we have to begin to talk about the various different types of material and how it's categorized. And there are plenty of examples right now that are being, shown with the different groups, like the COVID panels from Path and from what other groups are collecting at Find and elsewhere, that we should have and agree on a minimum data set of material samples and the kind of quality, how it's stored, and we have to sort of help people to do that. Maybe other panelists can also give comments on those, but I think we should at least minimally have the material, the description of how it's been tested, and how it's been stored and how it's being kept. Maybe other panelists can also make comments, and then asking the audience, for those who have recently been collecting materials, what have been some of the challenges and issues that you've had?
So maybe I can briefly share here my ideas. With regard to dengue and Zika, I know that these pathogens, there can be immunological interactions between them, and what has been sought after are special pathogens that might only have immune response against one of these pathogens and not against the other ones, because in the endemic areas, those viruses occur in parallel, also with chikungunya. So there could potentially be a hit list drawn up by scientists or by industry of the most sought after sample types. For example, people that were only exposed or infected with Zika and not before with dengue was maybe one of the more interesting samples that were hard to come by. And so that is on the pathogen side of things. And on the clinical epidemiological data side, which is kind of my area, it's important to have a standardized list of metadata that goes with the sample. Over.
Yeah, no, this has definitely been a challenge during the Zika outbreak. It was that Zika was circulating in dengue endemic areas, and the background of Zika sample is important. And now it's the other way around, right? We're in the process of evaluating vaccines for implementation, and we have prevalence studies to be run on Zika background.
And Jorge, you see that there's some comment in the chat about the freeze-thaw cycle. I think, could you or somebody in the panel address that?
Yeah, definitely. That's one of the things that we have observed is the the effect of freeze cycle samples, particularly in serological diagnosis, and that would be an important piece of information to collect.
Mm-hmm. And so thank you for that. I think, to me, the type of samples to share also should be, for instance, associated with outbreak or epidemic or a particular kind of situation, so that you can have them as a way to look at how things evolve, right? Because I think for our purpose here, the biological material of, pandemic and epidemic potential is important and to track. And I think one thing that also that Thomas Jaenisch mentioned is cohort data. I think some of the samples that should be prioritized would be samples that are cohorted, and there is intention to do serial collection over time. It doesn't have to be a very long time, it should be specified, but those cohort data can also help a lot. Maybe Thomas can speak to the value of those cohort data to have.
Sure, I'm happy to do that. So the value of cohort data is, for example, with associating certain clinical outcomes with biomarkers in the biological material. So the example here is dengue, so prediction of severe dengue. You need a large cohort because severe dengue is a rare outcome in order to have a few samples that were collected a few days before a severe dengue outcome occurred. So that means that those few samples that come out of thousands of initial samples that were collected are of high value because they may contain information that you can use for predicting severe outcome versus all the others who didn't proceed to severe outcomes. And I think we have a similar issue or challenge now with COVID-19. We want to know who is going to proceed to have long COVID and who doesn't, and are there any biomarkers that can be detected early on in order to associate clinical outcome with the biomarker that is predictive or that can be used as a prognosis. So cohort data allow these prognostic associations, which we are very interested in.
It also allows you to look at the decay of antibody titers through time and the specificity of antibody testing, particularly with flaviviruses.
That's true. And for example, in Zika, we saw that the many antibody assays became positive rather late in comparison to other flaviviruses. So it took like 10, 15 days until the maximum of the sensitivity was reached, which was not the case for the previous flaviviruses that we have investigated. So in order to see this kinetics over time, we need this prospective data collection.
Is there any other aspect of samples to be prioritized? How about, with Zika, there was a lot of interest about this during the Zika epidemic, and it was the different sample types where Zika was present. We found Zika in different body fluids that was different from other flaviviruses experience. Do you think that's an important sample type to be captured in our effort, or should we just focus on the serum samples?
So this is May. I would just say that we want to prioritize, some of the other samples, like you say. The serum samples are probably the easiest, and we sort of practice well with it. And I also think, like, in the effort that we're doing, we're really focusing on immunological markers and therefore looking at serum plasma, but a careful consideration must be made for other types of source materials, because they also may carry the biomarkers and may carry other things that, they prove to be more important later. And so I think one of the things is there is a comment from Mark Page on production of calibrated antibody secondary standards is not recognized. So we need to collect large volumes of that. Thank you, Mark. We certainly agree with you on that. And it's very important that the kind of samples we want to prioritize is what Mark says, is that at the very beginning of outbreak, we need to collect large volumes of materials that we can calibrate and then turn around and use them as reference material, and that enough of that's made and stored properly so that they can be shared elsewhere. And some of this can be sourced from a number of situations depending where the outbreak is. And we would then be collecting materials that may not be the emerging pathogen, but essentially to use for specificity typing of materials. So that's important. And let's see what else. I'm going to ask Tony, do you want to talk about the CREED?
Sure. So thanks, May. So, one of the things that we've been talking about inside CREED is exactly what kinds of specimens would we want to collect. And I agree with what May said, and I think what everybody here knows, which is collecting serum is probably the most straightforward thing. But limits you in terms of what you can do downstream. So we're having a lot of conversations about how does one collect viable PBMCs. I come out of the HIV space, so we're used to collecting PBMCs on a lot of clinical trials, and it's been incredibly informative. But it's an enormous cost to do that and to maintain a repository of viable PBMCs that can be used. It does bring incredible value, but it's a huge sunk cost, and one that you just have to make a decision up front that you can do that. So we're having a lot of conversations about that. And I think it's important to recognize that if you're going to collect, you need to also have control samples from the same population, because there are variations geographically based on likely genetic background, and also a tremendous number of environmental factors. And so, you don't want to get in a situation where you're comparing people from Durham, North Carolina as your controls and people from, Sao Paulo as your experimentals. The other comment I would make, and this is something that I've been thinking a lot about related to metadata, is all of those metadata values are going to have an error associated with them, right? If, vaccination status, are we talking about self-reported? Are we talking about someone shows you a card that may or may not be a real card? And I think having some discussion about exactly how good the data has to be, and recognizing there's going to be an error in the data collection process. It's not going to be perfect, and thinking about how to deal with that up front. So
I think also in reaction to your comment, Tony, I think it also is a nice introduction to the panel 3 discussion on sustainability. As you said, biobanking has a cost. And I think if we are talking in general about biobanking in times of epidemics and pandemics, as is the title of this workshop, I think it would be really useful at the very beginning of a pandemic to have a panel like this to convey and to just list a series of recommendations directed to the whole world, basically directed to all biorepositories who would have very interesting data or very interesting samples, but they don't necessarily have the expertise in biobanking or how a sample can be fit for purpose. So I think this kind of recommendation would be extremely useful, and it should be an ongoing task in the background of, I don't know, maybe WHO or other initiatives like that.
I couldn't agree more. And to emphasize that point, another big discussion that we've been having inside CREED is the idea of capacity building. Because of course, the only way that this is really going to work is if you have labs that are there that can do this, especially early on. And, since you don't know where it's going to hit, you need to be able to have expertise, in many, many different places. And how do you do that? And it's going to require a diversity of investment. I mean, if I had infinite time, infinite money, I could do that. But we have to get buy-in from so many different groups and get them to play nicely together, or at least agree not to fight.
So this is specifically important when we are talking about biobanking viruses and viral strains, as that's what we do in the European Virus Archive. And yeah, I couldn't agree more as well.
I'm going to ask Jorge to move to the next 2 questions. You have 4 minutes.
So the next topic would be the minimal quality standards necessary for this. What is in your view those minimal quality standards?
Yeah, I think we sort of discussed around that already, because Tony said, given the fact that there's going to be some inherent mistakes that we might be in the original metadata, but that we should specify that. And maybe those are some of the things that we can discuss in, as we collect samples and see what needs to be done, how to be shared. I would also ask that our other partners that's on the call, that Nicki, Tiffin, and I think Warren, I don't know if you're on from FIND, is that they are defining some of those already, and we can pull those in. Do you want to go to the next question and then we'll speak it for the sake of time?
And I just wanted to point out, so there are a couple of really good comments about the vaccinated individuals being useful for producing secondary standards, and
yes.
taking of sort of those comments on the side.
Yes.
yeah, the next question is to the sharing of COVID collections and how to make that accessible.
Maybe I can start a little bit on that, is that, you know, instead of Zika, which is rare samples and they're hard to get, and the cohort samples are very precious, we have millions and millions, maybe billion of COVID samples out there with about, you know, 100 some different collections and banks posted on the website. If you search for it, probably more. Is there a way that we need to sort of think about how to coordinate some of those or provide a method for some of those? Because I think as time goes, a number of these collections will be lost, and they may have some precious information or some use or utility to others, right? But that at a certain point, some of these will be discarded. Is there a need to talk about this, or this is not really our purview? That's really my question here. Does any of the panelists want to comment? So I think, Roger, let me put you on the spot. What are you going to do with your samples? Are you maintaining those, and how long that will be maintained? That's one of the questions.
Yeah, I think those are really important questions. And there are certainly issues that we're working around. Certainly as our partners contribute specimens to the repository based on consent, we may have varying timeframes where we can keep samples and when we have to dispose of them. There are certainly other concerns about, you know, just the pure age from, you know, whether it's a device developer or other researchers who are taking a look at, you know, if a specimen has been in a freezer for a certain period of time, are they going to, you know, trust it? You know, it may have come in with quality data when it came into the repository, but are you still going to trust it and have it as high quality and be a useful resource? So those are things that we all consider. And then, you know, for us, being a project-funded organization, we don't have an umbrella grant for maintaining repositories for perpetuity. So taking a look at the real costs of maintaining biobanks, and at some point, the number of specimens that we have left may not make sense to continue the repository. So do those specimens get discarded, or can they be transferred to somebody who has the infrastructure and make those available to be useful specimens?
Okay.
And this is again an issue where, you know, it has been brought up a couple of times in the chat, which is, you know, all of those issues surrounding the long-term detection of antibodies, vaccination status, and cohorts of vaccinees maintained, you know, maintaining of those samples through time. So what is your view of the value of the serum sample collections for COVID?
Jorge, was that directed to me or?
Possibly, or by anybody else.
Yeah, you know, we certainly have serum in our repository for COVID, but I will say the overwhelming interest is with nasal and respiratory specimens, and there are very few organizations that are really interested in taking a look at serology. Mainly our purposes and those that we work with are driven not by serology tests right now.
I'd like to make a comment about the length of sample maintenance, because it's a key point. And, you know, I'd said before, these aren't like wine. They don't necessarily accumulate value. But as Sarah Woodson pointed out in the chat, there is value to having things before and after. And I'll give you an example. I was lucky enough to get access to samples that came from CDC as part of the WITS cohort, which was the Women and Infants Transmission Study that was done in the early 90s in an era before antiretrovirals were widely available. And we were able to access samples from mothers and infants where perinatal transmission either did or did not occur. Now, that was a sample cohort where we got, serum and PBMCs, and it's something that could not be replicated today. It would be frankly unethical to do that study now. And we were able to get immunological insights by examining PBMCs that had been held for over 20 years, and they were still viable and still useful. So I think we should be careful about viewing things as, well, they're old, therefore they may not be valuable. Provenance matters. But there is a real cost to holding these things. I mean, there's no question about it. This is not cheap work. And back to the capacity building and the sustainability issues, if we really want to have value and we want to have snapshots into previous eras, we have to make an investment, and we've got to figure out how to do that. So.
But I think it's time for us to move to the next topic. I definitely agree with the need to balance our capacity to foresee the future needs versus the immediate need, like the example of the nasopharyngeal swab versus the serum. I think that with time, people are going to be interested in looking at parameters of protection and correlates of protection. So with that, I'd like to pass it on to the next session.
Thank you very much. So I think, we'd love the audience to put in questions and answers. I apologize you're not able to actively interact with our panelists, but our panelists are very rich and deep in experience. I'm asking them to jump in and make comments to these panels. The next panel would be guided by Thomas Jaenisch, and it will be specifically talking about solutions to barriers that we now have in accessing specimens and data. And thank you very much, Jorge. So
thanks, May, and thanks, Jorge. I just put up the guiding questions for panel 2. Let me read you the questions. So panel 2 is about identifying solutions to barriers that may interfere with timely access and benefits sharing to users and providers alike. And there's 3 questions we have prepared, but feel free to post additional questions in the chat if you feel that these don't cover the entire field. So the first question is, how should specimens and data be collected and harmonized? And this is an area that is quite close to me, as we are dealing with this on a daily basis now. And I would be curious what the audience and other panel discussants have to say about this. So I'm going to take notes here and move to the next slide. So this is the first question.
I'll make a comment right away, is that to me, from the very beginning, what we should do is be able to, this will be much more prospective than retrospective at this point because of the materials, and that prospectively, there should be a template that we agree on, and that we can also do many of the work ahead of time so that they can be collected appropriately. And that is a common template that everybody uses, with previous discussion on the minimal data set that we need to have. I think retrospectively, Thomas, you have the experience of talking about how to fit retrospectively, data on collections and how that has been a challenge.
Yes, retrospective harmonization of data is very labor intensive. There are several community developed standards like OMOP, CDISC, CDASH. I don't want to go into detail here because that's more on the data side of things. But there's also standards for the lab side like LOINC and SNOMED. So it would be great if there could be an agreement by the community, and if there could be some kind of meta harmonization or interoperability between the different standards, because now we have different standards that have been developed, so that if one group uses LOINC and the other SNOMED, that there's some algorithm that translates one into each other. Have any of the other panel discussants or from the audience, have you had experience with these standards and how to harmonize and standardize? Would you share that?
So I think the TGHN staff can make it available for our participants to speak instead of having to type in chat. Is that correct?
Absolutely. So if you raise your hand, we can unmute you.
Pardon? Please repeat that.
Zainab just said, please raise your hand in the big group, and she will unmute the participant. Okay. That is great. Thank you.
Yeah, so I see that Catherine Lowens had said something in the chat. Do you want to speak to this? Okay, if so. Yes.
Yes.
Yeah, okay. Yes, hello. I'm from University of Antwerp, and I'm involved in different European studies. And there we have a function as a central lab where we provide to all the centers that participate the lab material that needs to be used in these studies in order to provide a harmonized sampling collection. And we also provide and train the people that need to collect the samples also in order to get more harmonization on sample collection. That's what I wanted to say.
And is there a, so let me ask a follow-up question. Is there a particular way you collect? Is there a software that you use to collect that material, or they use that's common to it? And is that something that's proprietary or public?
Well, it's in European projects, and maybe from study to study, it will depend whether the manual is open to the public or only to the participating partners. That's something I need to check. I don't know, but yeah, I don't know.
Thanks, Catherine. Other discussants in the panel or in the...
Hi, yeah, this is Sarah Woodson. And I guess maybe I just wanted to comment on the way how I'm sort of thinking about this particular topic, especially in the larger context of a large virtual biorepository that would be, you know, hopefully accessible to many far and wide across the globe. But I think, at least in terms of, you know, sample collection, I think it would be very hard to actually harmonize this across the board, because pretty much all of the samples are going to initially be collected for a specific study and a specific use. And so, you know, I think in some areas, minimal, you know, sample collection procedures or standard sample collection procedures would be used, but not always. And so for something like maybe a virtual biorepository, an approach where you have, you know, like a collection of standard, you know, operating procedures or MOPs or something like that, that once somebody deposited something could point to and be like, yeah, we used that particular protocol. You know, that might be a good way to at least help link the information back to how the sample was collected and processed, or even through a publication. Because I don't know if it will be easy long-term to have any sort of real strict harmonization effort around that specimen collection. However, with the data, I think that part can be harmonized very easily, as you already mentioned, through templates and linking some of this additional information to it within the repository. Thanks.
Judith wants to have a comment, and after the comment, Thomas, let's move on to the next question.
In the context of, you know, low resource settings and LMICs, that really an approach where there is open source IT tools that people can use, otherwise homemade and very incomplete data will be collected, and if the data is not collected up front about the specimens going back is pretty much impossible. So having some open source software for specimen collection would be extremely helpful.
Thank you, Judith. Let's move to the next question. So the next question has the famous Nagoya protocol as one of the barriers. So what are possible solutions to legal and regulatory challenges that impede timely sharing, such as local laws, shipping, benefit sharing policies, like the Nagoya protocol? Are there any solutions that you have found that everybody could benefit from, that you want to point towards or show in this discussion? So the question goes to the whole floor, and please speak up or raise your hand.
Well, I think for some of the folks who have been shipping during the pandemic, I'm sure you have lots of insights into what has happened and how that can work. I also can probably give a quick update on what Nagoya protocol is. Nagoya protocol is very much a very high level discussion at the ministerial of health level, and that WHO is really taking the lead on some of this. But I think in general, the Nagoya protocol is in effect in over 100 some countries, which means that those ministries of health have to decide how they're going to meet the access to benefits sharing. And that's what Nagoya is principally about, is that if you receive materials from country X, then you will have to, in early on, make agreement to make sure that you're meeting that country X's Nagoya protocol has been adopted, and there is a policy in place, and that policy speaks to how you will share benefits back to them. Benefits back to them is something that we need to discuss, because benefit for one group is not benefit for another. So some of these are just simply benefits in access to training so that you know how to create a biorepository, benefits to access to research protocols, or benefits to vaccines and these things. And currently the Nagoya, the convention, what they call the Convention on Biological Diversity is meeting, and they're establishing perhaps a subset of Nagoya protocol that can be decided by public health, because public health is not included in Nagoya wording. And so the access to benefit sharing has been challenging to identify. So once they carve out, and if they do in this particular convention, a public health entity for Nagoya, then WHO can step in and sort of define what the benefit sharing access might be. So that's a long thing about Nagoya, but we do get that for people who says, oh, I can't share you because of Nagoya, but they themselves may not know exactly what Nagoya is for their country. I stop.
Thanks, but this is really good news that there might be updates to the Nagoya protocol that pertain to our field. Thank you, May. Are there other comments from the floor? Let's not limit ourselves to Nagoya only, but please, of course, share other stories around Nagoya if you have them.
Well, I'm just asking, does anyone in the audience have solutions to shipping and looking at some of those, because those are challenges too, very much so. Oh, no shipping challenges? Good, we don't have to deal with that, right?
All right, well, now that you've opened that door, so there are lots of shipping challenges. I mean, one of the big challenges that we have run into is that not every place has access to good or to reliable shippers, and you may have difficulty shipping things from one country to another. Even if they are abutted, you may have to do some sort of triangular movement to be able to get things to go around. And when you start talking about things that are under various degrees of regulations like select agents, that gets even more complicated because there are a lot of carriers that don't want to deal with that stuff. So one of the things we've been working on is trying to come up with just a list of like who could help, and then how, what parts of the world have access and how do we expand that. But no, it's an ongoing problem. And then, won't even get into the fact that a carrier may be reliable and then become unreliable. And what kind of backup plans do you have? So, unfortunately, I think that the carrier thing is an ongoing, you just have to keep looking at it and continually updating your information.
Do you think the virtual repository takes care of some of this, since you expect it's difficult from one country to another, but not a different combination depending on where the sample is sitting? Or would the problem be multiplied?
So if I may say, so at EVAD, so the European Virus Archive, of course, we do exchange viruses. So we are completely in this problematic with the couriers. And the option we took was to have one dedicated person that is in charge of the logistics. So basically it means that the person is kind of aware of all the problems, the various problems that there may be. And basically over the years, there is some kind of a background knowledge that is being built. So we try to issue documents for our network, providing directives or just guidance basically on how to do that. And basically we enrich this document every year or every month or whenever it occurs. And that's how we do it. So basically by experience, we would say.
Thank you.
Yes.
Should we move on to the next, or sorry, May?
Yeah, please move on to the next question. And then you'll have to close out in about 2 minutes.
So last question in panel 2, how to be responsive to the needs of different stakeholders, which has been mentioned by Judith in her talk, that we have done these expert interviews with academic, with commercial, and with companies and other stakeholders, such as ministries or international institutions. So how to be responsive to these different needs?
So again, I can maybe start. So at the European Virus Archive, we have approximately now 35% of our end users that come from industry. So that's because of COVID-19, of course. And basically we found out that the limiting step was the MTA and the time to access, because industrial companies are very in a hurry, are very much in a hurry to get the samples. Not saying like academia is not, but like for industrials, it's really the top thing. So basically we designed specific MTAs for different branch of our industrial end users. So for example, they would need our samples for testing antivirals or for testing diagnostics or for testing vaccines. And for those 3 applications, we have designed a specific MTA. So based on our experience, again, on what were the problems usually with the MTA negotiations, we generated those, I would say, more generic MTAs specific for some applications, and that speeds up the process. It's 35%, not 55%.
Oh, so sorry. Thanks for the feedback. Yeah, great. I realized that EVAG is really way ahead and really very professional. Are there other stakeholders that can share how they respond to different needs? Somebody from the big floor? Yes?
Hi, my name is Jaenisch from the World Reference Center for Emerging Viruses and Arboviruses here on behalf of Dr. Scott Weaver and Ken Plant. yeah, I gotta agree with Christine. Limiting the NTA specific to what the individual wants has been extremely helpful, as well as the emergency use agreement NTAs that we were getting at the start of the pandemic for COVID. Having an online form to help us screen through the various individuals, the requests that they wanted, what they were looking for, was it RNA, was it actual virus, et cetera. As well as having our own, again, dedicated shipping personnel definitely helps with the EHS requirements for our institution. On top of that, there's been a lot of previous talk about sample storage, shipment, et cetera. Another thing that we do that along with, that helps us with the shipping issues that we have, mainly lack of dry ice to certain countries, courier delays, et cetera. We freeze dry a bunch of our samples, those that we can get away with that. And that seems to help out a lot, but yeah.
Thank you very much. There are some comments in the chat. Maybe we can briefly have them discussed before we move on. Is that possible, May? There was a comment from Steve and from Roger.
I see Steve's comment. He's setting us perfectly for the next panel.
Okay, so, then let's move over.
Yeah, so I think what we can do is thank you very much for the comments here. And then let's move to the next panel to be led by Dr. Christine Pratt from EVAG. And you should get that started. So Christine, she's sharing her slides and we will get to this part. I think the benefits and sustainability is really key. Well, I mean, everything is key, but this one anchors the sustainability, how we keep going, right? So go ahead, Christine.
Yes, sorry. So yeah, this is a bit of an intimidating subject because how to cover that in 15 minutes. So I thought I would give a brief introduction on that just to start the discussion. It will be short. So some features of biobanks, they have many stakeholders as we have seen. The main products are usually not owned by the biobank, and the value of the products, it matures over a very long period of time. So basically, after 3 decades of biobank existence, it's been funded by public money for the large majority of them. And now there are challenges faced with budgetary restrictions. So do we have an idea of how many collections are actually closing down now? And one definition of sustainability could be the capacity of a biobank to remain operative, effective, and competitive over its expected lifetime. Some questions to find the causes of this financial threat that I mentioned before of many collections closing down. Is it a lack of publicity for the importance of biobanking? Is it a lack of accuracy in accounting for the benefits, monetary or not, arising from the use of biobanks? Is biobanking too expensive, et cetera, et cetera? So those are just a few questions that we have in mind. And determining the current value of a biobank is a bit complicated because the value is in its worth, the quality of the biospecimens, but it's also in its usefulness, the property of the cohorts. It's also in its importance, and there needs to be an assessment of biobanks as future assets. And what is the performance of the biobank? So as we have seen before, in various presentations today, value and value indicators can vary depending on the viewpoint, funders, institution, and users, it all depends. And the impact and utilization may be the most important. So the impact, it takes a long time to measure and it's difficult to track back to a single biobank. And the utilization is easier to measure and is influenced by several factors. So another problem that I wanted to mention is underutilization of biobanks. There's a recent survey that says that there are utilization rates of less than 10% for many, many biobanks. So maybe there are deficiencies in biobank design or model or biospecimen quality assurance, strategic planning, marketing,
I don't know, we can discuss that today. So here is a bibliography of really interesting recent papers on the subject, if you want to know more about that. And let's go to the question 1 of this panel. Can we identify the benefits and advantages for sample providers or for users? So I don't know if I can see the discussions. Yes.
So we'll also ask the TGHN to open this up to the floor. So those of you in the floor in the big room, if you have questions or experiences to share, please raise your hand. I'm gonna just put Steve on notice. I'm gonna come to you and ask you if you want to make speak to the comment you made. And then I'm gonna ask Tony Moody to comment on some of the things that Christine has said. So Steve, do you want to just mention what your thoughts are? You did a very eloquent chat in the chat. No. So if not, then Tony, do you want to comment on that 10% use of the biobank? We've heard that story from you.
Well, so I think actually what I want to start with is commenting on what Steve put in the chat, because this is something I've been going back and forth about with some of my colleagues here. I mean, one of the big issues that we have run across inside CREED is this idea of I'll speak from a US perspective because that's where I am, but I think it applies to a lot of other wealthy nations, which is we seem to come at this from a perspective of if we drop money into a location, that is sufficient. And I think we really have to go in with a sense of curiosity about what is it that will provide benefit to you so that we're not assuming that we know what our partners need in these locations. Is it that they need authorship on papers? Is it that they need intellectual property rights protected? You know, what are the drivers for this? Because I think it is far too easy to come in and go, well, I'm dropping in a bunch of money. That is an exceedingly colonial view of the world. And I think we have to get out of that. We have to look at this as a partnership. One thing that has come up inside CREED is a question of, you're going in and pulling samples from us. Why aren't you sharing samples back? You know, why aren't samples from where you are coming back to us? Why is this not bidirectional? Now, granted, you know, in CREED, we're looking at emerging infectious diseases. We're lucky in the US that most of these diseases don't circulate here, but it's a valid question to say, you know, what is going on here? Why do we not have a bidirectionality in this? In terms of, I mean, the investment for this is always going to be bigger than we think it is, right? It's always going to probably orders of magnitude bigger than we think it is. And I think we have to be very careful about thinking of sizing appropriately, but also recognizing we probably have to size up from what we initially think. And that there was a really eloquent story that was told actually during our recent advisory board meeting, that biobanking was very, very successful in certain locations until political unrest happened. And then all of a sudden you lost everything. So, I think we have to think about what are the drivers for people and kind of move that forward. So I'll be quiet now.
So Steve, I think you're off mute.
Yeah, can you hear me now?
Yes, we can.
Oh, great. Okay, thanks. Thanks, May. Yeah, I put that question up, to be a little bit provocative, but I think, one of the questions or concerns I have is that these conversations very quickly devolve into, and I'm purposely chosen that word devolve, into, publication credits and tenure and IP and all those things. And they're all important things. I don't disavow that, but I think we have to start from the standpoint of asking the people that are giving us the samples, what is it that you want? So, to follow on the point that was just made, not to assume, oh, okay, that Tony just said, not to assume that we want to just drop in a bunch of dollars and that's going to make everything fine, but actually say, what do you want from your participation? How does this help you? And I like Thomas's perspective of starting from the bottom up and trying to work in that manner. We're getting a lot of that challenge and question from investigators, particularly in low resource settings, who are tired of that unidirectional flow that Tony just mentioned as well. And so, we need to think creatively about how we can do this effectively. And I recognize it adds delay, but if we don't solve this problem, then it's just going to continually be viewed as exploitation of the people that are providing your samples. And you're hearing it from the ladies and the moms that are dealing with their infants in Brazil from Zika. It goes, as I mentioned, all the way back to Henrietta Lacks. You just kind of heard it a little bit from South Africa on Omicron, right? We identify it, share that insight to the world, and then we get shut down and locked down. And thank you for our participation. So I think I don't have any easy answers, but I think it's a really important thing that we have to start with, not do it, oh, by the way, I wonder what, but start with from day one. You're going to give me the sample. And that's true regardless of whether you're in Cape Town or whether you're in Cleveland. If you ask people, well, what would you like me to be able to do? And what would you hope would come out of your giving me this sample, besides your own diagnostic insight, but further than that? And I think we might be surprised by what we hear from those communities. Over.
So can I make a quick answer to that? And I think probably Judith Giri is going to say the same thing. So that's why we did that poll that Judith Giri presented as the last talk. We asked people, what are some of the benefits you would see? And Judith, why don't you comment on those who participated in that poll? And was more of a grassroots selection or opinion on what they wanted. And we thought we would start with that rather than saying, this is what you get, to do this. We also thought about at some point, maybe we can have an earned points system so that if you contribute, you do something, it's worth so many points, and after so many points you accrue, you could do X, you could, go to catalog and get X, Y, and Z benefits that they decide that these are the benefits they want. Over.
If I can add to that, this is Judith, for a minute. I think we did ask people, and not only on this project, but in other projects where we have done the same in low resource countries. And the answer is capacity building, give us training, build repositories for us. So the next outbreak, we can do this and keep the specimens and do some of the research here and do some of the development here. So that is, one of the things that we heard. Opportunities for networking, being part of the whole research community as another one.
Yes, I couldn't agree more with what you're saying. Basically, the problem sometimes is that, for example, when dealing with viruses, is that there are no BSL-3 facilities or level 3 or level 4 facilities in the country of origin from the virus. So there's no way the virus could be handled there. So basically, the capacity building is very critical locally.
I would add one point to the list that wasn't mentioned. Many, at least cohort investigators, are very attached to their cohorts. They want to be able to continue with their cohorts. And so basically they would say, let me just be able to replenish the samples that I give you by giving me funding so that I can continue this.
Yes.
I'd like to add one thing to that, which is one thing we've been discussing is the challenge of having samples come from a location where a pathogen is endemic, covered under rules at a biorepository that are very restrictive, and the fact that we have difficulty sending things back to researchers because we say things like they don't have the facilities, when in fact they are awash in this pathogen and have been working with the pathogen in their own laboratories for a long time. I think sometimes we let our view of regulations where we are locally get in the way of common sense. And I don't have a solution for it because I recognize it's a real challenge.
Well,
but you know,
yeah,
it's an issue.
In EVA, in the European Virus Archive, we have a specific board that is called maybe 2-3 times a year for those kinds of demands. For example, Mayaro virus is endemic in some country and like absolutely not here. And so we decide on a case-to-case basis if we are allowed to send the virus anyhow to an endemic region. And so we do have like a panel specialist that we can call to make some decisions. As I said, it happens very rarely, maybe it happened maybe 3 times in the past 4 years, but yes.
Just a quick, go ahead, Tony.
No, all I was going to say is that it seems as if you have a more advanced view of the world than we've got currently.
So just a more advanced view is the international health regulations at the WHO Annex 2A, what essentially says that if it's endemic in your country and you have the documentation that this is endemic in country, you may handle it X, Y, and Z way. If it's of unusual circumstance in your country, then you must then bump it up a bit. And so there is actually a guidance policy that can be looked at using that as an example. Sorry, I jumped in. I think
the past questions are nicely leading up to the second question, actually, that I'm going to put on the screen. So what are some successful models of equitable sharing and governance structure retaining local ownership? I think this is the previous discussion was kind of leading to that, basically. So the big terms here are equitable sharing and local ownership. So I'm just going to start with our model in EVA. So talking about local ownership, if a partner institute in EVA provides a virus to the collection, they do retain the ownership. But on top of that, if a country or if a lab in a country that is not part of EVA wants to give the collection to be distributed through EVA, to give the strain, the virus, to be distributed through EVA, we have what we call a specific MTA again. It's called a custody MTA. So it means that the providing lab, let's say in Cambodia or let's say in Cameroon, the providing lab is giving us the sample under a custodian MTA. So it means that they retain their ownership. It is clearly labeled on the online catalog and everything is submitted to them before we redistribute the results. So basically each time an end user from anywhere in the world wants to access to this resource that is just custody to EVA, we contact the originator and we ask them, are you okay to distribute it? And that's how it worked for EVA. So again, our solution was to have specific legal documents in place to be signed.
So I see Jorge's hand up. Is that still hand up and Judith's hand?
Yeah, I think Judith was before me.
Okay, then can lower hand.
My question is about, you know, is there any instances or examples where the collection, the sample biorepository entity provides support to the cohort from where the samples are coming in? And I'm saying that because a lot of times, and Thomas was mentioning the desire of the cohort PIs to continue with their cohorts at all costs. And sometimes if the cost of the sample, of obtaining the sample, which is actually, it's not that high to obtain a sample from a patient, just the cost of getting the sample, could be covered by the repository. Now you have a win-win situation where you see these two-way street, right? That we were talking about before, of the benefit for of the, you know, for the cohort versus the benefit of the bio collection and the user. And related to that, I have, in my experience, and this is coming from the CDC, we're not a commercial entity, but there is such a thing as a user fee, and I wonder what that experience is with your biorepositories. Those of you who have spoken about biorepositories, because a lot of times that resolves a lot of the issues. It resolves conflict of interest, research issues, where the user is simply paying a user fee for the samples that they need. It even resolves some of the shipping issues when that happens, because now there is a commercial value to the sample.
Hi, this is Sarah. I wanted to make a comment. So I think over the course of today, we've heard a lot about the agreements that go in place and ahead of time before the sample sharing, and what sort of agreements are made for ownership, and what sort of accreditation might be going back once the results are available and things like that. But what we haven't discussed, which might be an important topic, is the enforcement of those agreements and acknowledging, whatever acknowledgement instructions might be within the agreement. I think there's been several times where those agreements and that recognition was not necessarily honored in the most appropriate way, or the way that it was expected to be honored. And I don't think there's a lot of consequence for those actions when they do occur. So I think there might need to be additional discussion within the community about what are those consequences and of those types of actions. Thanks.
Yes, that is indeed a question that is difficult to solve, really. Like we do state in the MTA that the persons need to acknowledge in publication, but again, once the sample is out, it's a bit difficult to track everything down. Yeah, that can be a concern. So I've been told that I don't have a lot of time remaining, so I think we need to move on to the next question, which is, sorry. So what should be some key features of a virtual biobanking system to ensure sustainability? Vast question again. So maybe if some of the panelists or if some of the audience would like to start giving ideas on this specific question.
Yeah, maybe I can set this a little bit in context. I think there's a lot of models out there, and most of them are funding large bolus, either supported by the academia or the institution, and also by donor, philanthropies and so forth. And often those die over time if the interest wanes, right? And so I think one of the things that we've already referred to that Jorge mentioned is user's fee or cost recovery could be another model that balances donors and others. And also to encourage donors to pool resources for me to support particular biorepository activities, so that it's not the burden on one resource alone, and that it could be from a pool from which then there's a strategy that goes forward for 5 years or so forth to support. Because if you have it, and US government is especially subject to that. If you were just based on funding from year to year, it's very difficult to plan for the future. So either that or some kind of foundational system that is longer lasting can help very much. And a lot of the sustainability has to do with the purpose and sort of what we really want this for, and commit to long-term use potential, like Tony mentioned, for the early on days materials, because some of those are worth keeping, even though you don't know the purpose right now, they do come into use at other times. And how to decide that is, of course, very difficult and challenging, but we could also have, with so many biorepositories around the world, we could assign people to certain kinds of features and purposes that they have. That's another option. Okay, I speak too much, over.
So one of the difficulties again is how to ensure that your samples are fit for purpose. So that's one of the main, I think the main challenges in biobanking, purpose that is not known yet, as it will emerge in the future. And so basically there is a need to cross a line, to draw a line between hoarding samples and just having a collection that is useful. So it's always a difficult line. And I'm guessing that space is a problem for every biobank. Like we have limited space in freezers and we have limited personal resource to maintain the biobanks. So prioritization is also an issue. But I think one very important thing for biobank would be always to keep in mind to have stakeholder analysis in mind and to keep the different stakeholders informed about what the biobank is doing. Because it may be that in the future, the support will come from the donors. It might be patients association, they raise money and then they are able to fund you because they understand the work you're doing. It could also come from institutions such as, I don't know, public agencies or governments or things like that. But so basically having always in mind performance indicators and just like communicating to various stakeholders is to me a really key feature.
Okay. So thank you very much. We have 2 minutes left, 1 minute left, and I'm gonna beg your pardon and sort of take an extra minute or two to summarize this rich discussion. Thank you so much, everyone. Thank you to the speakers, the panelists, and the audience. So what we really like to do is to finish up this way and allow me to just share my slide one more time and we can take this out. Okay, so for me, the summary of the next steps is the key outcomes from this is that I think we still see many of the issues and problems and challenges are still there and we need to resolve them. And to me, the different working groups that we would like to set up, which is really about the type of specimens we want, looking at solving the solutions we've identified, and then lastly, trying to discuss deeper the sustainability and the benefits with respect to the source provider is so critical. And I think that we don't have all the answers at hand. What we like to do is, thank you and asking you to join working groups and volunteer joining the working groups. We promise that we don't make it heavy, but what we like to do is in the coming months, set up the 3 working groups and essentially meet one monthly or 2 or 3 times between now and the middle of next year, 2022, and come up with some statement and result and guidance. We welcome you to join our contact list. We cannot see you who have joined us here unless you opt into sharing the information. So we welcome you to go to our website, Biorepository at ucdenver.edu on the slide, and tell us what you thought and what working group you might like to join. We really think that having you join, this is really important. It's a grassroots effort, but we also know that from us, we can help guide and shape some of the other bigger initiatives you might have heard today, like the WHO BioHub, how we can actually synergize and work with them, work with other groups that are doing the same type of thing, work, and then come out with a plan that we can agree on and move forward. The biorepository area is very interesting because we all feel like we are archivists for the history of the past exposures, and we want to do our job responsibly. And we often feel we lack the tools and lack the credit or the strength to do it, right? But I think together we can make that much more productive. So again, thank you very much, everyone, for joining this. This is a sort of a journey that we like to take together. It's not finished yet, and there's much work to do. So please sign up and join us, and we will take it forward next. Again, thank you.